get everyone on the same page, the term that was raised in one of the questions, and again, feel free. We're definitely in the chatty part of the day now. So if you want to interrupt and ask for clarification on anything, or type in the Zoom chat, I'll be here. I'll be watching. But so the term that was raised was original anagenic sin. And this goes back actually to some papers from the 50s, I believe. And the person who coined the term was the son of a Presbyterian minister. And so it's why it has this sort of biblical overtone. And so the idea behind this phenomenon was they were looking in the mouse model of influenza, and they were infecting mice with two closely related H1N1 virus strains. And again, this is the 40s or the 50s, and so that's kind of all we had. And this is the classic definition of original anagenic sin, where you vaccinate with one virus and you generate antibodies here that are specific for that virus, the light blue virus. And then you come in with a strain that's kind of closely related. And this is the key thing in original anagenic sin for it to work, is it has to be closely related enough that these light blue antibodies bind at some level to the dark blue HA protein here. It can't be totally different, or else this phenomenon won't happen. And as a result, you get these light blue antibodies that are binding, that are recalled, and you don't make very many new dark blue antibodies. And so the sin is this original imprint of the light blue response. And the key thing in this mouse model is that these light blue antibodies, when faced with the dark blue HA infection, didn't work. And so the mice did worse than if they had never seen the priming virus. So that's the big key, is naive animals did better than immune animals, because this immune response was actually inhibiting the production, this memory immune response was inhibiting the production of a productive de novo immune response. And so partially because of I think the drama of the phrasing and the cleverness of it all, and the interest in the actual model, this became like a big thing people have talked about, not just in flu and HIV and dengue and many other systems, but actually demonstrating it happening in humans has been incredibly difficult. And even in mice, it doesn't happen very often. And the best model for it is still that original model. It was almost as if this was a little bit of a consequence of the ideal situation with the two strains that were picked. Now, there was some evidence in the sort of mid-2010s that this was starting to happen in humans, getting the H3 vaccines. And the H3 viruses at that time were, for influenza, were drifting in a way that you had this kind of situation where antibodies, where strains would be closely related, but potentially to have non-neutralizing cross-reactive antibody responses. And so what probably limits this from being a significant phenomenon is you need to have lots of antibodies that are enough to remove an antigen from priming a new response, but not effective enough to actually provide you any immune protection. And that, I mean, even saying it, it's kind of a contradiction. If it's enough to mask the antigen so that it doesn't prime an immune response, it should mask the antigen in enough of a way that it should provide you with some protection. So what there is evidence for is maybe not this sort of, you do worse than if you had not seen the vaccine before, but you don't do as good as you could have done if you hadn't seen the vaccine before, which is a different claim. The vaccine still provides some protection, but it's not as good as if you had just been vaccinated directly with the dark blue strain. That there's clear evidence for, and I hope that I didn't butcher that too badly. So you can definitely get a less good response than you would have if you were seeing the antigen for the first time, but it's probably not going to be actively harmful, and it will be helpful, just not as helpful as it could be. This is probably to some extent what's happening right now a little bit with SARS-CoV-2 is you have antibodies. They're not the greatest antibodies you could have if you were magically able to get a vaccine that perfectly matched the strains that were circulating right now, that would definitely be better. But it's not going to hurt you to get the original vaccine. Those antibodies are less than ideal, but they're not actively harmful. But that's classic OAS. It comes up constantly, and people raise it a lot. But I would say the leaders in the field, this paper was written by Florian Kramer, who's an antibody flu and coronavirologist, don't really believe it's a significant contributor to human pathogen dynamics. The other problem with the term is it gets applied to any kind of imprinted immune response. And it really only refers to the situation where a priming event actually makes you worse off in a challenge. And so to contrast it, he introduces these other terms that are also used, but provides really strict definitions for them. So there's also a term called back boosting, which is I got vaccinated with the first strain, the light blue strain. Then I see the second strain, and I actually expand my response to the light blue strain, but I also make a great response to the dark blue strain. This is very common. This is happening all day, every day with the coronaviruses. So when you get infected with SARS-CoV-2, let's call that the dark blue strain, you expand a bunch of antibodies to other coronaviruses like OC43, NL63, and so forth, which are more like the light blue strains. They're actually pretty far apart in the antigenic space, but there's enough overlap, particularly in the spike protein, in the conserved S2 domain of the spike protein, which is sort of like this bottom part. This is HA, but they look kind of similar. So the stalk part of spike is also conserved in the same way that the stalk part of HA is kind of conserved. And so you get this sort of cross-reactive recall of unrelated memory that doesn't necessarily help you very much, but it doesn't hurt you at all. And it doesn't do anything to actually limit the boosting of the de novo response. Okay, so that's back boosting. And then there's head epitope specific imprinting. Oh, I should say, some people hate this term because it's really just cross-reactive recall. We didn't necessarily need a special name for it, but it is useful shorthand when you have these situations like we have with Corona, where there's all these multiple strains. And when you say back boosting, people now can kind of know you mean this kind of situation. So for flu, and probably for SARS-CoV-2, we have this this issue with as you prime and boost and challenge with multiple flu vaccines, or you get multiple flu infections, what you see develop over time is these very specific antibodies to epitopes, so the regions of the protein that the antibodies are going to bind that are in specific areas of the protein. And so for HA, this is sort of the structure of HA is like a lollipop. This is the part of the HA that binds to the sialic acid, and that's going to mediate the entry into the cell. And those antibodies that are head specific are the ones that have the chance to be the most potent. And so they're going to block that interaction. They're also the most exposed to the immune system. And so as a result, the more you see those types of epitopes, the more you expand that response. So you end up, if you look in somebody receiving their very first flu vaccine, they tend to make this very balanced response across the whole HA. And then if you look at someone 20 years later that's seen multiple flu vaccines and multiple flu infections, their antibodies are much more biased towards head-specific antibodies. And we think that's just that exposure of the antigen that just makes it easier and easier and easier for the immune response to see it, where it's maybe a bit harder for the immune response to see these stalk antibodies and to see the stalk region to make stalk antibodies. The thing about influenza, and the reason, I mean, it's not surprising, it's a well-adapted, highly evolved pathogen, is it can change these head epitopes pretty easily, and it can't change the stalk epitopes pretty easily. So it sort of sets up the situation where it shows you the part of the virus that it wants you to make the antibody response to. For that particular strain, it's going to be neutralizing, and it's going to stop you from getting that strain again. But for the species as a whole, it's better for flu if you focus on the head, because these stalk antibodies are, in the long run, going to prevent you from seeing any flu virus. And so that's led to the fixation recently on generating stalk-specific antibodies. This is a similar thing in HIV too. HIV focuses the immune response on the most variable part of GP120 because it knows it can get around that, even if that particular response ends up being very protective for that particular strain. There's some flu-specific types of imprinting that I think it's probably not worth going into too much, but there's broad categories of the different flu HAs. So we categorize them as group 1 or group 2. And H1 viruses happen to be in group 1, and H3 viruses, which are the other circulating human viruses, are in group 2. And some of the avian flus are either group 2, like H7, or group 1, like H5. And what has been found there is if you were first exposed and have very good memory to H1 viruses, you appear to be more protected against H5 viruses, versus if your first exposures were to H3 viruses, which means you were born after 1968, then you are more likely to be protected against H7. And it might explain some of the epidemiology of who's been most susceptible to these avian viruses in areas where there's been avian crossover. And so that's basically all I wanted to talk about here with the these different forms of original anagenic sin. Antigen masking is basically a modified form of this as well. Well, actually, this is worth talking about. So here, the idea behind antigen masking is I give you the influenza vaccine, and I say, and maybe some of you have to get the influenza vaccine every year, right? You work in a hospital, you have to do it every year. And, somewhere about your 5th or 6th time getting the influenza vaccine, you enter a study and they measure your response to the influenza vaccine. And they say, you actually didn't respond very well to the flu vaccine. Your baseline titer prior to the vaccine was basically equivalent to your post-vaccination titer. And that's what this is looking at, is you actually already had a lot of antibodies to that vaccine. So when you saw that exact same vaccine, which happens with flu, or a very similar vaccine, again, there was going to be no new priming of new B cells because the antigen from the vaccine was immediately coated with the antibodies you already had, wasn't made available to the B cells, so there was no boost. And so as a result, it looks like the vaccine didn't work, but it's not that it didn't work, it's that you didn't need it. And there was no way to know that you didn't need it until you actually got the vaccine. So that's antigen masking. And that actually, I'm glad it's on here because that's going to be relevant to the paper. So these are all a bit different. So this is all sort of related to this prior antibody affecting subsequent immune responses and exposure to the immune system. There's another kind of prior antibody interaction that's medically important, and that's this idea of antibody-dependent enhancement. And the primary virus that this is seen in, and really the best studied case of it, is in dengue virus. And so dengue is another flavivirus, and has a surface envelope protein, and it has 4 strains that are distinguished by this E protein dimer on its cell surface. And these, you can think of these like serotypes, like you would see in flu, or in flu, and so they're sort of very distinct strains. And what's been observed in dengue infection is that your very first dengue infection is usually a relatively mild infection that is respiratory and sort of systemic in nature. Your second dengue infection with a new strain, because you don't get infected with the same strain twice, it's like you get sterilizing immunity. Your second infection can cause a hemorrhagic fever. And so when you hear about dengue hemorrhagic fever, it's always your second exposure. And there were varying theories over the years as to why this was. And one of the competing theories from the animal models was that it was actually a result of something like original anagenic sin, but it was this antibody-dependent enhancement. And so the idea was you made antibodies in your primary infection. You also made CD8 T cell responses, which were protective. If you saw exactly the same strain again, the antibodies neutralized that serotype. You had seen it before, you don't get infection. But if you got a new serotype, the antibody bound to the virus, but didn't prevent it from doing its job. The virus didn't get prevented. And in fact, the antibody brought the virus to a dendritic cell, which is where dengue viruses like to replicate. and facilitated the virus getting into the dendritic cell. And so what this did was, rather than helping get rid of the virus, it actually helped the virus get to a reservoir. Again, this is almost a little bit like HIV flavored, where the virus has gotten an express route to the cells that would ultimately kill it, but now it's also the cell that it's going to grow in. And so this then causes increased immune activation and cytokine storm type effects. And so this was actually a controversy in the dengue field as to whether or not this was happening. You could show it in vitro, you could show it in mouse models in vivo, but you could also show other mechanisms of hemorrhagic fever-like syndromes in vivo that didn't rely on antibodies at all. And so it had been a question for a number of years as to whether or not this was really the mechanism that was driving the response in humans. And relatively recently, 2017, the data came out from a very nice cohort study that really showed that it probably was this antibody dependent enhancement that was driving the hemorrhagic fever and shock syndrome. And so this is a simple figure sort of showing that, looking at antibody titers and binning people based on whether they had very low antibody titers and then increasingly high antibody titers. And the people that develop shock syndrome and hemorrhagic fever, which is a very, very clear specific phenotype, you can't mistake it for anything else, were the people with sort of average levels of antibodies. These sort of like, they're above background. These people you would call negative. And these people you would call like, yeah, they've got some antibodies, but they're not a lot of antibodies. But once you had a lot of antibodies to any strain, any of the 4 strains, you didn't get shock syndrome. And so there was this sort of narrow window in which some level of antibody was bad. I wouldn't call this original anagenic sin in the sense that it was blocking a productive immune response. Instead, it's this effect where you're actually facilitating viral replication. And this just shows similar data. Again, it was lower antibody levels that actually drove this particular phenotype. This is there's a lot of modeling in this paper, and it's epidemiological modeling, but I think if you're interested in this type of topic at all, I think you would enjoy this.